let's investigate some chemistry of life. We're going to talk a little bit about, well, a lot about biomolecules. We're going to discuss a couple specific chemical reactions, the hydrolysis and dehydration reactions, and then we'll talk a little bit about how water is so special and important for biology. So it doesn't matter what kind of organism you are, if you're a person, if you're a beautiful flower, if you're a baby porcupine, or even the patoo, my very favorite bird. I will have a link to it put up because it is 1000% ridiculous. It looks like a Muppet and it lives in Jamaica. All of these living organisms have something in common. They have several things in common, but the one we're going to talk about today is that they all have in their chemical makeup 4 classes of specific molecules. These are the biomolecules. They are required for life, and I suggest if you're setting up your notes for this part, it's helpful to set it up into a quadrant system so you can talk about each thing, each specific characteristic of, say, the carbohydrates, the lipids, nucleic acids, and proteins in kind of the same order to help you compare them. But we're going to talk about these. We're going to talk about the carbs, the lipids, the nucleic acids, and the proteins, and how they're similar but also how they're different and why they're so essential for life. So these are all organic compounds. And remember that an organic compound is something that it has carbon. So in lay terms or in marketing terms, that something organic has a specific definitions, but the definition we care about is the biological one, which is that it is a carbon-containing molecule. The organic compounds are generally polymers, and we're going to talk about what that means. Lipid is the exception. Polymers are they're chains of molecules, or they're molecules that are made up of smaller molecules. So it's easy to think of this if you think of a arts and crafts necklace, like something made out of smaller parts, like a macaroni necklace you might make at camp or something. So if you consider the individual pastas as monomers, so there's one, there's one, there's one, there's one, etc., and you put them all together, this is a polymer made of multiple monomers of macaroni noodles. Okay, so monomer means one, poly means many, and when you put a bunch of monomers together, you make a polymer. So we're going to see that come up consistently in this lecture. So, again, so another way to think of this, or like the technical way to think of this, is that the carbohydrates, the proteins, and the nucleic acids are made of repeating subunits. So the lipids are a special case, but all the others are going to be made of, you'll have some specific unit, and then you'll subunit, called a monomer, and then you'll see it get repeated over and over. Now these could also be different flavors or different specific kinds of monomers that get put together. We'll see that come up too, and that's actually really important. So let's start with the carbohydrates. So your carbohydrates are your sugars. You may be on a carb-free diet, and again, this is a term that means something different in lay terms or in the marketing or real world than it does in biology and anatomy. If you're on a carb-free diet, if you're avoiding carbs, you're basically avoiding grains. But you can't avoid carbs and stay alive. You need carbs to survive. So a carbohydrate include the sugars, so these are your simple carbs, and the starches, which are more complex carbs. So anytime you see a word that ends in OSE, that is a carbohydrate, okay? So examples being glucose, which is a very simple sugar, fructose, which we find in our fruits. We have sucrose, which is table sugar. We'll talk about how that is a polymer. Ribose and deoxyribose are found in DNA and RNA. Those themselves, ribose and deoxyribose by themselves are sugars, but they are used to make something different we'll talk about. Cellulose is found in plants. It makes up the cell walls of plants. And the carbs that we know and love, such as potatoes and pastas, are a bit more complex. But whenever we eat things like that, we break down any sugar or any compound containing sugar. If we're going to store it, we store it as glycogen. So glycogen, and there's a lot of GL words happening here, you just kind of have to go with it. Glycogen is how we store energy for quick use. And that's the basic function of carbs, is it's quick use energy. So glycogen doesn't last very long in the body. You can store glucose as glycogen in your liver or your muscles for about a day, and then you run out. If you don't eat anything, you burn through that energy. So we have a long-term molecule also that we'll get to in a second, but let's start here. So carbohydrates have a very distinct structure in that they have these circle structures. Okay, so when you see anything that has this ring structure, it's very likely a carbohydrate. Now, it's easy to draw them as this kind of honeycomb looking shape, but in real life they look a bit more like this when they're three-dimensional, but that's very hard to draw. But you can still see that repeating ring structure. Boom, boom, boom. Okay, so especially if you have starch, you'll see each one looks exactly the same and repeats like beads on a string. Okay, so we have this nice ring structure. So if you look over here at this starch, we actually have an enzyme. So an enzyme is not a carbohydrate, but this enzyme specifically takes a starch, like maybe you have a piece of bread, you put it in your mouth, and you just let it dissolve. When you let a piece of bread dissolve in your mouth, you'll notice it starts to taste really sweet. And what's happening is this enzyme amylase is breaking down the polymer of starch into its component monomers of glucose, and that's what you're tasting. You're tasting that glucose come out. So if you break down the word carbohydrate, you're seeing that it's made of carbon, hydrogen, and then oxygen. Just trust me on this. So the chemicals that make up the carbohydrates are carbon, hydrogen, and oxygen. Importance. That's why I mentioned it. And another important thing is not only can you identify carbohydrates by their ring structure, but you can also look at their chemical structure like this. And anytime you see the hydrogen, you look at the number of hydrogens and you compare it to the number of oxygens. So in cellulose, there are 10 hydrogens and 5 oxygens. And since there are twice as many hydrogens as there are oxygens, I can automatically identify this as a carbohydrate. Okay. In glucose, we have 12 hydrogens and 6 oxygens, so that's a 2-to-1 ratio. Carbohydrates. That's just a nice, easy way if you're looking at the chemical structure or looking at the formula to identify something as a carb. Hint, hint. So we can take the monomers of carbohydrates and string them up. That sounds weird. We can take the monomers of carbohydrates and we can attach them with chemical bonds. So you can put multiple ones together. And every time you make a bond, if you're bonding two different sugars, the technical term for a sugar is a saccharide. So this is a saccharide bond. So if you have two sugars together, that is called a disaccharide. If you have just one sugar, that's called a monosaccharide. You can also have a whole bunch of sugars connected, and that is going to be a polysaccharide. So these are just terms you need to make sure you know. Okay. So examples include if you have sucrose, which is just table sugar, that is a disaccharide, so it's made of two sugars, and so each sucrose molecule is glucose plus fructose. Okay, cellulose, on the other hand, is a long chain of many D-glucose molecules. So don't worry about what that means. Just know that it's a whole bunch of the same monomer over and over and over, which gives plant cells their rigid structure. So the point of carbohydrates, the function of carbohydrates, is just to give you quick energy. So sugars are a quick, simple energy, okay? So if your blood sugar is low, you can eat a candy, drink some orange juice better than candy. And I had a friend once who said, oh, my blood sugar's low, I'm going to go drink a diet soda. That's not really going to work as well because that's not a usable sugar. It's not the same kind of sugar. That's why diet is what it is. But the basic function of carbohydrates is to provide a quick sugar, okay? So at least the simple sugars do. These starches are a more long-term sugar. It takes a bit longer for that energy to be broken down into something usable. So a lot of times you see athletes before a big game, you can tell I'm from Texas because my original example is from football, they will, before a big game, they go and they just carbo load. They just eat a whole bunch of pasta or they eat a whole bunch of bread. And then they get out on the field and they have so much energy. And then in that case, carbo loading is a really good thing. The problem, though, is that our bodies are basically always, just through evolution, they're always ready to just store energy away in case we need it, just in case we have to run away from lions. But there aren't a lot of lions just hanging out in San Francisco, so when we eat carbs and our body stores it, if we don't use it right away, it ends up getting stored as something else, and we'll talk about it in just a second. Okay, here we are. We're going to talk about right now, which is lipids. So lipids are the next class of biomolecules, And they include the fats, the waxes, the steroids, and fat-soluble vitamins. We talked a little bit about something about lipids in the last lecture where we said that they are not water-soluble, they're hydrophobic. So whenever you think about things mixing like oil and water, that means they don't mix at all. So this is the oil component of that. So the lipids are hydrophobic for the most part, and they include stuff like butter, olive oil, and cell membranes. So we'll talk about what that means right now. So the structure of lipids, very normal. Well, the first kind you ever learned about is the kind where you have this one backbone, and then hanging off of it are 3 fatty acids. So we have a glyceride. Okay, so the glyceride is the backbone. So there's that backbone, there's that backbone, 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 and then you have the fatty acid tails hanging off of it. The fatty acid tails can look different, but that's the basic structure. Now, sometimes that one of those fatty acid tails can be replaced with something else, like a phosphate group, and that's what's happening up here. So you have your 2 tails, which are being attached to that glycerol, and a phosphate group on this side. Now, the tails, the fatty acid tails, are hydrophobic, and so they're going to move away from water. Water cannot dissolve them. But the phosphate side can interact with water. It's hydrophilic. So when you have a whole bunch of these phospholipids hanging out together, they'll self-align, they'll self-aggregate into the cell wall. So that's what's happening here on this cell. You see all these little dots with legs on them. Boop, boop. Those are those phospholipids, okay? And they're aggregating so that the inside, all of this, are those hydrophobic sides of that fatty acid, so they don't want to interact with water. And the outsides, on both sides here, is the phosphate side, which is hydrophilic and does like to interact with water. And we're going to see when we talk about osmosis and diffusion that that is incredibly important. But here's some other ways to identify lipids. Lipids are also made of the chemicals carbon, hydrogen, and oxygen. Okay, so carbon, hydrogen, oxygen. So how can you identify if you have a lipid versus a carbohydrate if you don't have the structure? Well, in the case of the lipids, the hydrogen and oxygen ratios are very different, okay? So it's not a 2-to-1 difference like we see in the carbs. So anytime you don't have a 2-to-1 ratio, so you don't have twice as many hydrogens as oxygens, you likely have a lipid if it's just got C, H, and O in it. Okay, so we're going to see some examples of that in just a minute. So there is no monomer of the lipid, okay, because they don't aggregate into long chains. They don't bond in long chains like the carbohydrates do. They might hang out together. They aggregate in those phospholipid bilayers, but they don't bond to each other like a true polymer would, okay? So that is a big difference here. But again, in each case, we have that glycerol backbone and then those fatty acid chain tails. Okay. Sometimes those fatty acid chain tails have a little kink in them. And so this is the difference between a saturated and an unsaturated fat. So a saturated fat, all the tails sit very nicely. Okay. So first of all, let's talk about what that means. So not the sitting nicely part. But if you look at carbon, like we talked about in the last lecture, carbon has 4 valence electrons, which means it can either get rid of 4 electrons, it can steal 4 electrons, or it can share those 4 electrons. In any case, it's got 4 things it can do something with, 4 electrons it can bond with. And so it makes 4 bonds. Now, if that carbon is saturated, that means that it's got a bond to as many hydrogens as it can, and then a carbon, okay? So it's bonded to, on the end, it's bonded to 3 hydrogens. In the middle, it's bonded to 2 hydrogens. Anytime instead of a hydrogen, so like this could have a hydrogen here, this could have a hydrogen here, but instead you get what's called a double bond. So anytime you have a double bond, you get a little kink in your structure because it's a stronger bond, and so the whole thing kind of bends a little bit. Now, the saturated fatty acids can sit nicely, okay? So you have this one sitting nicely, and then you have another one sitting right under it, and they lay very flat, and they usually create solids, or very close to solids. So that's going to be your butters and your lards. So these are very high energy. There's a lot of energy going on in there. And so when that happens, there's a lot of energy that can then be stored later well, it gets stored as fat, is basically what I'm trying to say. Because we aren't working out to the extent that our ancestors were in trying to survive, if you eat butter and lard, it gets stored as fat it just gets stored in your body very quickly. Unsaturated fats, on the other hand, are going to be liquid at room temperature, and that's going to be your olive oils, anything that's an oil. I can't think of another example right now, but those tend to be healthier for you. All right, so what's the function of lipid? So a lipid is going to be a long-term energy storage molecule. So anytime you have so carbohydrates, you use them up really quickly, and if you don't use them, they get converted to lipids and they're stored as fat. So even if you have fat-free gummy bears, if you have any sugar in those gummy bears, if you don't burn off all of that energy within a day, that sugar then gets converted to fat. So it might start out as fat-free, but you end up accumulating fat. And if we were on the savannah and trying to hunt for our food and doing all these crazy active things all the time, this would be great because there's so much energy in these things and we need that energy to stay alive. But as overfed humans, we tend to take in more energy than we use up, and instead we store it as fat. So, how do we distinguish our carbs and our lipids? And I'm sorry about this, I'm not really sure what's happening with this thing. Go here. But let's see. So, we know that both of them have CH and O as their molecules, their atoms within them. Both of them store energy. Carbs store energy for quick usage, while lipids store energy for long time usage. It can be stored for a long time. And we know that if you have too many carbs, you have excess carbs, that gets turned into fat. So how do you distinguish between a carb and a lipid? And what this weird formatting issue is trying to tell you is there's two things. You can look at the H to O ratio. So do you have twice as many hydrogens as oxygens? If you do, then that is a carbohydrate. And you can look at the molecular structure. So if you have something with that ring shape, it might be a carb. Okay, so really it's kind of boop, 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 something like that. Okay, if you have something that's got a backbone and three things hanging off of it, that's going to be a lipid. Now, there's a lot of variation in this, so you sometimes can't just rely on that structure. And instead, you look at the hydrogen to oxygen ratio. So if you have some molecule and you look at its hydrogen and you say, oh, there's 12 hydrogens, and there's, that looks like there we go, and there's 6 oxygens. Hey, there's twice as many hydrogens as oxygens. This is a carbohydrate. If, on the other hand, you have something else, and I don't know if this is a real molecule, I'm going to make it up. Oh, look at that. This is not 2 times 5. It's more than that, so that must be a lipid. Foop and foop. So let's do some practice. So, if you were to look at this molecule, and I'd hope this would pop up in a second, but it doesn't, whatever. So how would you know that this is a lipid? So this structure is not the traditional glycerol with 3 things hanging off of it to make a nice happy lipid, but I can look at this hydrogen and I can say, hey, I have 34 hydrogens. If this were a carbohydrate, I would have half as many oxygens. I don't. I have 2 oxygens. So this has to be a lipid because it's not a 2-to-1 ratio of of hydrogens to oxygen. So it is definitely a lipid. This guy, I could look at either the structure. So I have a nice ring structure here. Okay, so there's 2 different ring structures together, so it's definitely not a lipid. But I can confirm this by looking at the hydrogen and the oxygen. So since I have twice as many hydrogen as oxygen, this has to be a carbohydrate. All right, there are rings. There are rings, so this must be a carbohydrate, right? Ooh, let's double check. I don't have a 2-to-1 ratio. If you don't have a number here, that means it's just, we consider it a 1, okay? Because why would we put that oxygen there if there's not at least one of them? Just to make sure we're on the same page, this 27 only refers to the carbon, the 46 refers to the hydrogen, and the 1 would refer to the oxygen. So there is definitely not a 2-to-1 ratio here. It's 46-to-1, so it's not a carbohydrate. These are actually the wrong kinds of rings. They are doing something different, okay? So they don't count for ring structure, and this is actually a lipid. This is technically cholesterol. Another thing that people say they have to avoid, but actually without it, you wouldn't survive. So there's that. What about this one? So this structure doesn't make any sense in our traditional lipid sense. Where's the carbon backbone? Where's that glycerol backbone? What's happening? I'm going to have to instead look at my ratios. Okay, so 24 divided by 2 is not 2, it's 12. If this was a 12, I would say it's a carb, but it's not. And so this must be a lipid, and it is. All right, so I have a nice happy ring structure. But, you know, that last one burned me, so I better check my ratios. 10 divided by 2 is 5, so this is, in fact, a carbohydrate. Okay, so that was carbohydrates and lipids. Those both store energy. Carbohydrates do it short term, lipids do it long term. If you have excess carbohydrates at the end of the day or whenever, when your body's dealing with the stuff, they get moved into fats. Okay, they get moved into lipids. But let's talk about proteins. So proteins do so many things, and they're so important. We're going to come back to the fact that they're enzymes in a little bit. But these include, proteins are found in meats, they're found in dairy products, eggs, and some plants. So you can be a total vegan and still get enough proteins, but it's a little harder. You have to make sure you're getting the right things. So you have to make sure you have a diet high in nuts and beans, other things. Tofu is made from a bean, so it's made from soybeans, so that's a good choice. We'll move on from there. So the structure of proteins is incredibly varied. Proteins are so important and they do so many different things that there's no way you can just say, all proteins look like this. But you can say that they all start as a complex chain of linked amino acids. Sometimes a functional protein might just be 1 or 2 amino acids. So we're going to talk about what amino acids are in a second, but they're very important. But most proteins are made of a whole lot of amino acids. So what's happening here, all these little dots, each one is an individual amino acid. And they get arranged in a certain way. So they get put into a chain in a certain order. And then that chain gets arranged in a certain way. And then it starts interacting with other chains in different ways. And then finally, it's a functional protein once it's all completely put together in the exact right way. Okay, so but you can see different proteins have different structures. So insulin, which is really important for making sure your sugar is going to where it needs to be after you eat, is just a couple of polypeptide chains. That's it, and then it's functional. On the other hand, catalase is so many polypeptides, and they have to be arranged just right, just perfectly, so that there's this one teeny little spot where the thing it's going to be acting on can get in there and do something, or will have stuff do to it, whatever it's doing. But so these two proteins are very, very different, okay? But the one thing we can say about, we'll get there in a minute. Okay, so what are proteins made out of? So remember in our last lecture we talked about how the most important elements to life are CHNOPs, so CHNOPS. And we've already looked at how lipids have carbon, hydrogen, and oxygen. Well, proteins have nitrogen and sometimes sulfur. So the only thing they're missing is that phosphorus. Excellent. So that's important to remember. If you see any chemical formula that has a nitrogen in it, it's likely not a carbon or a carbohydrate or a lipid. The H to O ratio is not relevant in this case. It's not applicable because proteins are so complicated. All right, so the monomer of a protein is called an amino acid. And it's interesting because there are only 20 amino acids. So there's basically an alphabet of 20 different letters, an alphabet of 20 amino acids that have to be arranged in so many different ways to make up all the proteins. And there are hundreds, millions perhaps, there are so many proteins. We do not know how many proteins we have yet. We're still discovering new ones every day. There's a protein that's only job, as far as we know right now, is to make sure when you're developing as a fetus that your right hand is exactly right. So your thumb is basically your hand isn't upside down. So it makes sure your hand, as you're develops so that the palm is on the right side and not the wrong side. And sometimes that protein doesn't work, and people are born with their hands inverted, and they have to go through surgery as a really young child, a really young infant, to get their hands fixed. It doesn't happen often, but that's the fault of one protein, as far as we know, that's actually causing that. So proteins are incredibly important. And so it's that sequence of amino acids that makes proteins what they are initially. Okay, so another word for protein, well, basically the adjective of protein is peptide. And so if you have a string of amino acids, then that is called a polypeptide. So you could have a dipeptide, that means there's just 2 amino acids. But once you have more than 2, it's called a polypeptide. Every time you have an amino acid connected to another amino acid, it's connected by a peptide bonds. So then once you have a whole bunch of them together and you start folding them, and then they start interacting in a certain way, that is called a protein. So here's an example of here are the 20 essential amino acids, so you have to have them survive. We make all but 9 of them, and so the 9 that we don't make, we have to take in our food. So for example, tryptophan is an amino acid that we don't make, but it's essential for us to survive, and so we have to eat it when we eat food. We have to make sure when we're eating food, our diet includes these 9 essential amino acids that we don't make, which are these guys. I in no way expect you to memorize these. This is just a table for you to see that each one of these is very, very different. But the things they have in common are the end groups, okay? So they all have this same end group. And really, they all have this same end group, but sometimes it's hard to find. This one actually, tyrosine, looks like it doesn't have it, so it's complicated, but really you can see that they all have this carboxyl group, or they all have that carbon-oxygen combination on that side, and they have this amine here, so that's what this nitrogen is. And this is why, so urea is a protein. The reason why urine smells is because urea, when you are peeing, the nitrogen of it breaks off. And is that what it is? There's something about that interaction in urine where the nitrogen comes off, and that's what smells, and that's the smell of urine. I'll post a link to something that talks about it. Okay, so again, amino acid chains are connected where each polypeptide, between each amino acid is a polypeptide bond. This is not a polypeptide bond. This is a sulfide bridge, or a sulfur bridge. And what that does is it just helps the amino acids come together in certain ways. So this usually happens at cysteines. That's not important for you to know, but it's just interesting. So the reason why there's not one here is because it only happens between specific amino acids, and those aren't the specific ones. Okay, so what the heck is the point of proteins? So proteins have 4 major things that they do, or really 3. One is they provide structure. Okay, so actin is in our muscles, and what it does, if you look down here, is it has these little, and we're going to talk a lot about this, so it has these little finger-like projections, basically, and then when a contraction of the muscle happens, it pulls these together this way, the other proteins, the myosin proteins, and so that's part of the structure of the muscle. We'll get into that in much more detail later. We're going to skip to proteins are also really involved in repair and maintenance of your body. So they're always going around and fixing things, because everything in your body is always breaking down. It's just kind of the way life works. But on the other hand, everything in your body is also being built up. So up to a certain point, it's being built back together, because your proteins are going around and making sure everything is doing what it's supposed to be doing, or getting to where it's supposed to be. So one of the biggest functions of proteins is this catalyzing reactions thing. And so what that means, so imagine you're at a party and you see 2 people and you know they would be great together, and you're just like, man, I really hope they meet each other. And they're just randomly moving through a party, and they're not really getting any closer to each other, and you're just sitting in the corner like, just come on already, just talk to each other, oh my god. And so you go over and you grab one of them by the hand and you take them over to the other person and you say, hey, this is blah blah. I really think you guys would hit it off. Whatever it is that you need to do to get them to talk to each other. Well, you just catalyzed a reaction. Because sure, maybe randomly those 2 people would have bumped into each other at the party, but it could have taken a long time. So by you intervening, you catalyze that reaction by making it happen faster. So you stepped in and lowered the amount of energy required from the system to make something happen. All right. And so, sorry, weird message. Okay, so that's what an enzyme does. So an enzyme is a protein that lowers the amount of energy required for a reaction. Now, the idea of proteins acting as enzymes, or being enzymes that lower activation energy, is so important that we're going to come back to it in a few minutes. So let's move on real quick. So we're going to talk a little bit about the nucleic acids next. So the nucleic acids include one of the most famous molecules that we know about, DNA. And so we're going to spend just a little bit of time talking about DNA today, and we're going to talk about it more later. The nucleic acids also include RNA. And so if you have a book, and it's your very favorite book, and you know that someone wants to borrow it, the likelihood that you're going to let them borrow your book, if you know that and this is a book, by the way, if you know that there's somebody who, if it's the only edition of this book that exists, and you want to make sure that it stays perfect forever, you're not going to just lend it to someone. You might make a copy of it so that they can borrow it. Maybe they'll borrow the copy. So the book, though, would never leave your home because you want to make sure it's safe forever. That's kind of like DNA. DNA has all of your information, all of your genetic information in it, but it stays in the nucleus. So this is a cell, and this is the nucleus. The DNA never leaves the nucleus. It stays in there. It stays nice and safe. And instead, if you need a gene to be active, to be used, the whole point of the DNA being there, you use RNA instead. So RNA comes in, and we're going to talk about what it does. And it basically makes a copy of the active part of the DNA that you need for the cell to function. That's really all you need to know about the big difference between those two right now. We're going to talk a little bit more about it in a second. But basically, DNA is the book, and RNA is the working copy. So DNA and RNA are found in all living things, just like all of these these biomolecules. In the eukaryotes, which include the algae and the protist, the fungi, the plants, and the animals, this is found in the nucleus of the cell. We'll talk more about organelles later. In the bacteria, so here's a bacteria, there's no nucleus, and so instead the DNA just is free floating. And the bacteria are such simple organisms that they have such simple DNA in comparison to the higher organisms that it doesn't matter if their DNA gets damaged because there's always going to be another bacteria. They make copies so quickly that they aren't really worried about it. Alright, so nucleic acids. Let's talk a little bit more. So some nucleic acids are only a single strand, while DNA, we know, has their very famous double helix strand, or double helix structure. Okay, so that it can either be single-stranded or double-stranded. Another difference is that the subunits of DNA and RNA are slightly different. They both use A, C, and G, and we'll talk about what that means. But DNA uses T, while RNA uses U. So what these are, if you look over here, they are the monomers of DNA and RNA. So the monomers of the nucleic acids come in 5 flavors really. Okay, so RNA only uses these 4, while DNA also uses thiamine. And this is a horrible way to write it, but you don't need to know exactly what it's called. But DNA, instead of using uracil, uses thiamine. So, and over here is an example of how these work. So there's T, G, C, A. So T will always bond with A through hydrogen bonding, which we talked about last time. So remember, hydrogen bonding is the weakest form of bonding that we're looking at. And so if a single hydrogen is bonded to another hydrogen, it's so easy to break. But if you have a whole bunch of those, it's like having a lot of Velcro. It's harder to pull those two ends apart, and so it becomes very effective. But the fact that it's really easy to break individual bonds is really important because that means you can open up the DNA for it to be copied. Okay, so nucleic acids, if we're looking at CHNOPS again. CHNOPS. So we've said that the lipids and carbs have CHO. The proteins have nitrogen and sometimes sulfur. Well, the nucleic acids have CHNOP. So they have everything, so they have everything except for sulfur. And so the important thing here is that phosphorus. This is incredibly important. This is a very high energy molecule. Well, it makes very high energy bonds. It's the energy currency of the, we'll get to that in a minute. We'll get to that in a different lecture. Again, nucleic acids are so complicated, if you look back over here, that we don't care about the hydrogen to oxygen ratio. So that only matters in the carbohydrates and the lipids. So the monomer of nucleic acids is called the nucleotide. So you're kind of seeing a theme here with nucleic. Okay, there we go. So the nucleotide is made of a phosphate group. So just like how we saw a phosphate group in the phospholipid bilayer, there's phosphate involved here. Okay. There we go. Okay, and so it has a phosphate group. It has a nitrogen base. So that's these guys. So they're called nucleotides. And then, that's not a nucleotide, that's the base. So that's an A, T, C, G, and U that we saw. Okay, and then there's a sugar. So DNA stands for deoxyribose, and that ose makes it a sugar. RNA is ribose. Okay, it's ribonucleic acid. DNA is deoxyribonucleic acid. So there's a sugar involved here, but because it's involved in something very, very specific, we don't count this as a sugar. It's not functioning as a sugar. It's not an energy molecule. So what is DNA for? DNA is all about heredity, or nucleic acids are all about heredity. So if you have a non-functional gene, you're not going to be able to make the protein you need for some function. So, for example, in very light hair, what's happening is you don't have the protein that makes your hair darker. That protein is not functional, and so instead your hair comes out light colored, or so it's lacking the protein that colors it. Okay, so where that comes from is first you have a gene, okay, and then that gene is read and becomes, so this is DNA, and then you have RNA, and then that goes to make amino acid chains, which then lead to the expression. Okay, so the expression of, if you start with your genes, and then you get to your proteins. And we're going to go into this in much more detail later. But the basic thing to know is that your nucleic acids store all of your genetic information. They code for proteins. So those four nucleotides are arranged in certain orders so that they can code for the amino acids to be put in a certain order. And it's the expression of protein, so the presence or absence of a protein, that causes a genetic trait, like something you might see or some kind of functionality. So let's, I'm going to skip this. I'm going to make you do this as part of the homework. Can you figure out what these are? I mean, the answer's there. We might as well go through it. Just kidding. Okay, so which of these, so pretend this, look at the answer after you figure it out. If you are given these clues, can you figure out what biomolecule we're looking at? So the rigid structure of plant cells, maybe that doesn't help me, but as a long chain of monosaccharides, you know a saccharide is a sugar, so this must be a carbohydrate. What about if you have something that the clue is genes and CHNOP? That would tell you you're looking at a nucleic acid. If you're looking at a structure that looks like this, and you've got anything not hydrophilic, but hydrophobic, whoops, I'm circling the wrong thing. If it's hydrophobic, the only hydrophobic thing we've really talked about are the lipids. Okay, this must be a lipid. It also makes up cell membranes, and so that is lipid. What if you have something, the monomer is a nucleotide. You've got that nucleo root, so it must be a nucleic acid. If you have a macromolecules that's huge and complex, that almost always is going to tell you it's a protein, not always, but that amino acid right there as the monomer, connected by a peptide bond, these are words that should make you think protein. What about this? It ends in OSE. That automatically should tell you carbohydrate. Even if you don't look at the rest of it, that tells you it's broken down into simple ones, and look like this happy ring structure. Oh, that has to be a carbohydrate. And again, that OSE will only ever be used on words that tell you it's a carbohydrate. So what about this? This tells us almost nothing. What is this? Does this structure tell us anything? But we can look here and say, oh, if the hydrogen to oxygen ratio is more than 2 to 1, so that means it's something like 3, and half of 3 is 1.5, but oh, it's only a 1 for the oxygen. That means this must, and then you can pick any kind of example of numbers here. So if it's more than 2 to 1, you know you must have a lipid and not a carbohydrate. So let's take a couple minutes to talk about enzymes. So remember that enzymes are proteins. They are biological catalysts. So biological catalyst means it's a biomolecule that is being used to speed up a reaction. And what that means is, so there are catalysts that are not biological, they're just acids or something, and they make a reaction go faster or slower. They only make them go faster. Heat also can make things go faster. That's important there. But an enzyme is specifically a protein catalyst. Okay? So these are very substrate specific. They're like a lock and a key. So any slight variation in a protein shape is going to change what can be put into it and used. And we'll see an example of that. So these two are not the same. They can't make the same thing happen. Okay. One important thing about, and we're going to come back to these words, activation energy, in a second. One very important thing is that enzymes are reusable. Once your enzyme goes through and it does something, like if we go back to the amylase in our spit, in our saliva, once it breaks down that big old sugar into simple sugars, it then can go and act again on some other sugar and make that do something. So they get used over and over and over. So unlike the sugars, which end in OSE, the enzymes end in ASE. So we'll see examples of this in a second. And they are affected by temperature and heat. So, temperature and heat are the same thing, sort of. For our class they are. pH. So pH is the presence of hydrogens, and we're going to talk about this in a second. But temperature, so a fun example of why temperature is really important is, so your immune system functions through a lot of enzymatic activity. So a lot of enzymes are present to keep you healthy. People used to think that you got colds because you were cold. And so if you were getting sick, they'd say, put on a sweater. And then people figured out, no, it's not that you're cold. It's that there are viruses and the germs are making you sick and you're not eating well. And it's just nothing. It's not the cold, though. What a bunch of baloney. And so people were like, oh, obviously it's not the cold. It just makes you feel better to feel warmer. Oh, but wait, recent evidence suggests that, well, yes, viruses do cause colds. And if you're healthier, you're less likely to get a virus. You're more likely to fight it off. But being cold slows down your immune system. And so enzymes and other proteins don't function as well when they're really cold. They don't work as quickly. On the other hand, they also can break down if you have really extreme temperatures, like really hot temperatures. So if you ever had a medication or if you ever had anything that says, you must refrigerate or store at some certain temperature, it's because if it's not in that temperature, if it's any hotter, then it's going to break down faster. There's a lot of medications where it'll say store at 70 degrees, store at 52 degrees, or whatever it is, and it's just trying to tell you if you leave it on the counter, if you leave it in your car, it's going to go bad because the enzymes break down and then can't function. So this is a molecular structure on the left of each member of these dots. Each one is a specific amino acid, and they're all put together a certain way, but that's really hard to draw, so we like to use this kind of schematic model where we draw this little Pac-Man looking guy, and we show that only very specific substrate can fit into that enzyme, into the active site, and make something happen. So once this is put here, so this is called the lock and key model, and it makes sense because you have a specific lock, and it's only going to be able to take a specific key to make something happen. Once that key is in the lock, the enzyme can do its work. So it might break it down, it might take it to another spot, it might put something together. There's so many things that enzymes do. But the one thing they all do is they speed up reactions. See how I've said that like 8 million times now? Maybe it's important. And they do this by lowering the activation energy. So it's kind of like if you're looking at someone on a hill and they really they know that once they get a rock rolling, it's going to roll further. So you only have to give it a little bit of energy to get it over that hill. But wouldn't it be easier if that hill were lower? That would be so great. You wouldn't have to push as much. It's so much less energy you have to put into it. And that's what an enzyme does. So in this example, if you had some sort of magical unicorn claw, something that can just take the entire top of this mountain off, or like dynamite, I guess that's a better example, then that would be like an enzyme that would be acting as a catalyst to make this go faster. So in terms of chemistry, if you have two things, and if you put them together, you're going to get two new things. Okay, so that's a basic chemical reaction. Well, there is an in-between state where they're in the process of BC handing off. So basically C is going to hand off B to A. So there's a point where they're all connected, and that's called the transition state. So when that happens, it can happen faster if you have an enzyme making that happen. So it's basically facilitating that reaction or facilitating that process, that transition state, so it happens faster. Because these things might happen completely randomly. Like that rock might completely randomly be pushed up the hill by wind, But how likely is that to happen? You don't know. Probably very unlikely. So what the enzyme does is make it happen much more quickly. And sometimes it just makes it happen at all. It may not happen at all without the enzyme present. Okay? So one thing to remember is the more enzyme you have, the faster the reaction's going to go. And that's just, if there's way more people helping out with someone roll their rock over the hill, they're going to get that job done faster. So same thing here. The more enzymes you have that will randomly hit the products, the more, the reactants, which are on this side, the faster you'll get the whole thing to the product side. So enzymes are substrate specific. And so substrate is just whatever it is that the enzyme is working on. Okay, so what that means is if you're not lactose intolerant, what happens is you have lactase, so it ends in ASE, which means it's the enzyme that works on lactose. Lactose is a sugar found in milk, and lactase takes that sugar and it breaks it up into two parts, galactose and glucose. However, sucrose, which is table sugar, cannot be broken down by lactase. It sits in it awkwardly, it doesn't work, and you have to have the perfect fit to make that enzyme function. So in this case, you can see that the substrate does not fit the enzyme, the active side of the enzyme, so it's not going to function. So another way that so this is an example of what an enzyme might do. So an enzyme might say, I'm going to take this thing, and I'm going to take this thing, and I'm going to put them together. And when it puts it together, if the two ends, if this is like a Lego piece, and you have this Lego piece getting stuck on this Lego piece, but you have to kick out these extra pieces because they're extras. My Lego analogy is getting really weird now. But basically, if you are trying to make two chemicals come together, and as a result, you have a hydrogen end and a hydrogen and oxygen end sticking together, what's going to happen is water is going to get kicked out, and then you end up with a bond here as a result. So these two things normally wouldn't do anything. They're just chilling on their own. But once they come into contact, they make a bond between themselves, which is water. They release these bonds, and then you end up with one final molecule. Now, because water is being released, this is called a dehydration synthesis reaction. And so if you've ever dehydrated vegetables or meat or something, you know that the water has to come out of it. So you're dehydrating it by taking the water away. On the other hand, the reason why pH is very important is because, and this works just with water too, their hydrolysis is where water breaks things down. So lysis to break. And so what water does is if you have a polar or you have a hydrophilic molecule and you have a bunch of water around, it goes and it's like, dude, I'm just going to break through this. Boom. And it breaks it up by splitting itself into hydrogen and oxygen and hydrogen and then causing this original bond to break. So that's a hydrolysis reaction. It's the exact opposite of a dehydration reaction. Now, this can also happen with just a whole lot of H's present. And if you have something that has a whole bunch of hydrogens present, that is a high pH solution. If you have something with a whole lot of OHs present, that's a basic solution. If you have a whole bunch of H's present, that's an acidic solution. And these can also go in and disrupt these bonds. So that's why enzymes can be less functional if your pH is not properly set, if your pH is not correct. We're going to talk about how this becomes really, really important in breathing later. So let's talk about water. We talked about water just a second ago. What's the deal with water? Why is it so important? Why is it that when we look at other planets, one of the very first things we look for is water? And the reason why we exist, one of many reasons why we exist on this planet, is because water is such a unique molecule. It's very polar. So oxygen is a certain size, and it has two extra electron pairs that are hanging out by themselves. Just kind of taking up space on this side, and because there's electron pairs, they're very negative. So they give this side of the molecule a very negative feel, or negative pole. Whereas on this side, since the electrons are being pulled in this direction, the hydrogens have a bit of a positive pole. So they have a positive side. But that means that you have it's basically like a magnet with a north and a south end, and they can attract each other. So you can get a bunch of water molecules stacking on top of each other. Okay, and you can also get them on this side. So it'll be the opposite way where you have the oxygen. Okay, so it's attracted to that hydrogen, and this oxygen is attracted to that hydrogen. Okay, so the polarity of oxygen is incredibly important. The opposite sides have opposite charges. That means that water can stick to itself, and that is called cohesion. Whoops, sorry. Water can also stick to anything else that is hydrophilic, and that's called adhesion. Okay, so that's important. And so anytime if you have let's say you have a wine glass, and you see, you swirl your wine, and you see the legs, I think that's what they're called, where you see the water kind of streaking up the side instead of just falling straight down. It's streaking up the side, or streaking down the side over time, because it is sticking to the glass, because the water is sticking to the glass. Another example is if you have a glass and you see the water kind of does this, which we see all the time, and so you may never think about it, but what's happening is the water is bonding to the glass, the side of the glass, and kind of creeping up a little bit. Now, if you put adhesion and cohesion together, you can get capillary action. So if you look at a plant, and this is a close-up of a plant, let's make this green. So here's our plant. Okay, here's a leaf. Yay! Here are some roots in the ground. So a plant is going to pull water up, and it doesn't do it actively. So it's not like the plant like strains a muscle and it pulls some water, because they don't have muscles. So the only way to get water to move through a plant is you have the water evaporate out of the leaf, and then all the water molecules are basically in the giant chain. It's like a bunch of happy hand holding. And as soon as the first water molecule leaves, it pulls the next one into its spot, and then it pulls the next one into its spot, and it pulls, and it pulls, and it pulls, and it pulls, and then the whole chain moves up the plant. And then you can move water and anything that's dissolved in the water from the roots to the leaf. And that is called capillary action. We're going to see capillary action in some of our labs. Okay, another important thing about water is that it has surface tension. So at the top of water, so if you have a water molecule, and it looks kind of like this, hydrogen, hydrogen, and it can bond to other water molecules. So here is an oxygen, hydrogen, hydrogen, boop, boop, boop. Okay, so then this can also bond to another hydrogen on another water molecule, and so we can see that you really get 4 bonds, because this hydrogen can then bond to another oxygen. Oh my god, they all associate so closely. At the top, there's less to bind to because there's no water up here. Okay, so at the top of the water, the surface of the water, these water molecules are bonding to each other more strongly. And I actually did that backwards. Boop, boop, boop. Okay, so it'd be like if the hydrogen is here, and then oxygen. So they're basically putting more energy into the bonds to each other at the top. So these bonds are a lot stronger, which means if you belly flop onto this water, it's going to hurt a lot because you have to break more bonds than you would if you were just swimming through the water. So that's surface tension, and that's really important. There are animals that can walk on the top of water, and that's really important. There's also, water does something where the bonds between the water are really strong, okay, and the bonds within water are really strong. And so because of the bonds, so we'll talk first about heat of specific heat, which is how much energy does it take to raise the temperature of your object 1 degree. And it turns out water is very, very hard to heat. So you may think it's easy because you make a lot of pasta, like I do, but really the fact that it takes so long just for water to boil, no other chemical does that. They all happen really quickly. Water takes a lot of time because it takes so much heat to be added to it so that it will start to go into the vaporization phase, which is this one. So what that means is if you live next to a body of water, you know it's a lot cooler than if you live somewhere very far from a body of water. And why that is because you have all this heat coming in, and it's going into the water, and it might be coming off of the land, so this is land, and it's giving off heat. Also, all that heat's going into the water, but it's not heating up the water. It just goes into the bonds between the oxygen and the hydrogen and just gets stored there. And it takes a whole lot of that energy before those bonds break. Another important thing is to think about how ice is less dense than water, which means if you have an ice cap, if we have ice caps left, they float on top of water, which is really important. So first of all, that if you have ice on top of water, it means that something can live underneath it. Okay, so when water freezes, I've never lived anywhere where water totally freezes, but when that happens, if water was, and I don't know what I'm drawing here, let me go back. So if you have a lake and it freezes over, but that lake is also full of fish, if ice was more dense than water, this ice sheet would crush those fish underneath. Oh no! Also, the entire lake would freeze solid. It would be really hard to deal with. But the case, what actually happens is that ice floats on top of the water, the organisms underneath barely notice, everything is fine, and then this actually stays warmer than the air above it because it has this insulating ice layer now. Yeah, that is actually, and the reason why this happens actually is because as water starts to get in order so that it can be a solid, it spaces itself out a bit more than it would as a liquid. Okay, so that is all. I mean, that's plenty of information for this module. Please make sure you try those review questions, and I will see you in lab. Bye.